Preeclampsia is one of the most serious complications of pregnancy. It's clinically characterised by severe or high blood pressure in the mother and protein in her urine. It affects around 8% of all pregnancies, and it claims the lives of around 60,000 mothers each year and far greater numbers of her babies. So in preeclampsia, we think there's an early defect in the establishment or the setup of the placenta, and this leads to poor placental blood flow to the baby. And it's this hypoxic or this really stressed state that the baby then and the placenta starts to release antioxidant toxins into the mother's bloodstream. And they spread around the mother and they start attacking her vital organs and her blood vessel system. And so, unfortunately, there's still no cure for preeclampsia. The only treatment is to remove the dysfunctional placenta and with it the baby. And this can be terribly detrimental if the baby is premature. So you can imagine that the goal for many of us in the research field is to try and come up with a therapy that's safe in pregnancy, that will block these anti-blood vessel proteins, and also then help to protect the mother's vasculature, or the vessels, from being attacked by being pregnant. And so we set out to see if we could actually block these toxic proteins or toxic factors being produced from these placentas. And so we generated some small ribonucleic acids, which you don't need to know too much about, but they're very similar to DNA, but much smaller. And they're able to bind and inhibit gene production in the dysfunctional placenta. But that's great. How are we going to get it into the placenta? So we've got these tiny little things called nanoparticles. They're like a bubble, if you will, and they're stable. They're made out of a phospholipid bilayer, so all that is a bacterial cell wall. The surface of the bacteria is stripped, and we're able to put into those bubbles tiny amounts of drugs, such as chemotherapeutics, or in this case, gene silencing agents to block the production of toxic factors in disease settings. And so how do we get them to the placenta itself? We have to target them some way. So these little nanoparticles, or these biospheres, are decorated in these specific targeting antibodies that will basically home these vesicles to the placenta to unload the cargo or the drug so that we can have directed therapy. And so we've chosen to do that via a receptor that we know is 3,000 times higher expressed on the human placenta. And importantly for us, it's produced on the surface, or the side that's in contact with the maternal blood. So we're able to get good access to the placenta to deliver high loads of these nanoparticles that should be able to then quench the toxic factors. And so in the lab, what we do is we dissect out human placentas, we take tiny pieces, pop them in a culture dish, which is all very exciting, and we apply these nanoparticles that have these gene targets, or they're able to quench or block the production of these toxic genes in the placenta. And so what we do is we flip these upside down, allowing these placental pieces to float like they would in the body. And this allows the nanoparticles to migrate and find the placenta and actually unload and deliver the drug or the RNA that we are trying to put in to block these toxic proteins. So you can see from the graph on the left-hand side, you've got our control, and on the right, basically, it's these targeted gene silencing agents. And we're able to block the production of this gene, but importantly, we're able to also show that we can block the actual release of these proteins or these toxic factors that would go out into the blood. And so what we're now doing is we've been working with mice to see what happens in the body. And so we put placental targeted and non-targeted nanoparticles with these gene silencing agents in, and we're able to deliver them into the mice. And then we collect the placentas of these mice at the end of pregnancy, and we can have a look to see if we've been able to deliver these agents into the cells, not just the surface, but actually into the placenta itself, where they'll have the greatest effect. And so we've been able to do this, and we can find that we get really good delivery when the placenta was collected from animals that had these targeted approach, and you can see on the image on the left the red fluorescence shows that the actual sRNA, or the gene silencing agent, has managed to get into the placenta. And you can see on the right where we've got non-targeted, we do see a little bit, but we've got much less. So we can see we're really enriching the delivery of these blocking agents to quench these toxins of preeclampsia. And we're now testing this in our mouse model of preeclampsia, where we're actually able, by viral techniques, to induce these mouse placentas to overexpress or overproduce these toxins of preeclampsia, just like in women. And we're able to do this through IVF techniques and then transfer these embryos back. And so what happens is, just like in women, we get these mice that have only high production of these toxins that we see in the clinical situation. And we also see a nice or a steep increase in their blood pressure, just like we see in women. So we're really excited by this concept, because this offers potentially the first opportunity for us to actually target placental delivery, so that we don't get off-target effects in the baby, we don't get off-target effects in the mother, and we can deliver much lower doses. But it also extends beyond preeclampsia, because there are many other diseases or problems out there where a dysfunctional placenta is at play, such as fetal growth restriction and miscarriage. And so we hope that this research not only will provide the proof we can actually target delivery to the placenta, but we can find novel treatments for these terrible pregnancy complications that are claiming so many babies and mothers each year. And so I wouldn't be able to do any of this work without a huge team behind me, and many of the innovators in this room will agree. So I just wanted to make sure I acknowledge the team, the amazing lab staff that we've got, and the scientists, the clinical team, and the patients that donate these tissues to us, as well as our collaborators, and of course the funding bodies that support this work, and without we wouldn't be able to do. Thank you.